Now, the applications and implication of this whole study of microbial diversity, or whole study of genome and genome diversity, is basically help you out to utilize these microorganisms and to make use of these microorganisms in future. Suppose if you identify some microorganisms and it has been identified under certain class, group or categories, and you have marked it as the, say for example, species X. And that species has some characters which can give you better quality wine for wine production. Now, until unless you mark that species, that this is the wine producing species, right, and you categorize them into certain class, order, group, or in any hierarchy, finally species level, right, that would be easy for the globally, it is easy to identify that marked strain to use for the purpose of the wine production. So this is how the identification help in application of this. So how you translate this whole study, this is translating whole understanding about the microbial diversity and categorizing them to placing them into different classification, different categories in order to use in future. So just we talk about different diversity in environmental and industrial contexts, how this diversity will help you to understand the industrial application and the diversity in the environmental conditions we'll discuss now. Now just talk about the composting ecosystem. Now the composting ecosystem, the microbial succession in composter system demonstrate how different microbial communities sequentially break down the organic matter. Now, if you take the ecosystem of composting any organic matter, and you have to identify what kind of microorganisms involved in that, which bacteria is better to decompose these flora or these organic matter. Or for example, in case of oil spill, or in case of any organic matter where you need to decompose it, right. So it is easy to identifying these bacteria from such kind of ecosystem through whole genome analysis, through metagenomics, if they are non-culturable microorganisms, and to establish that, okay, this organisms involved in decomposition of a particular organic matter. So this is one of the advantage of this. And this soil is rich in this bacteria. Similarly, environmental bioremediations, how to remediate the pollution, water pollution, how to rectify the different organic matter which are polluting the water bodies, maybe ocean, maybe rivers, it may be any water bodies where the animals are also living. So if you are contaminating the water bodies through your oil spill, through your organic matter, through pollutant, then the fishes, the animal flora in the whole bodies, water bodies will be affected. So in order to that, you have to identify what are the different species which are better utilized to these organic matter, these oil spills, and to completely degrade them. Now the specialized microorganisms to be identified, which degrade the environmental pollutant, environmental contaminations, maybe hydrocarbons, maybe heavy metals, right, maybe some synthetic chemicals. So these natural cleanup process, right, offer sustainable alternative to expensive physical process. For example, physical process means you have to make some physical method for purification of rivers, oceans and water bodies. But this is something which can be, very naturally can be utilized and give you the remediate the help in remediation of the water bodies and restoring the methods and provide you the sustainable methodology for maintaining the environmental situation or environmental pollution. Then come to the agriculture enhancement. What is the role of these microorganisms in the agricultures? Various beneficial soil microorganisms which are the symbiosis, means we have a good relation between the plant and the soil, and these microorganisms which can improve the plant soil, which can improve the plant health, which can improve the nitrogen fixation, right? And these microbial inoculant can be utilized to to provide to the plant in order to get the good crops and good yield of the plants. So this is always good that compared to chemical fertilizer. Chemical fertilizer can be harmful for the plants, can be harmful for the consumers like human and animals when they consume the plants, vegetables, fruits and all. So in order to avoid these kind of a contamination, in order to avoid this kind of a harmful effect, the microbial flora can be well utilized, and this provides the sustainable agriculture practice while enhancing the crop yield. So the question comes, how do you know which microorganisms to be taken to provide the sustainable agriculture growth, sustainable environmental bioremediation, and compositing ecosystem, decomposing the organic matter? Again, the answer of the question is that you should go to the whole genome analysis of the microbial world, metagenomics if non-culturable identified, and even simple sequencing if they are cultured in the laboratories, and identify them, mark them, and place them certain group, class, and order. Systematically, you classify them for the further use. Similarly, medical and biotechnological frontiers. What are the medical and biotechnological frontiers? The novel antibiotic discovery is again the microorganisms. The whole microbial world basically providing the best quality antibiotics, the best quality antimicrobial agents in the form of molecules, in the form of organic molecules, in the form of peptides, and many more. So, and these can be used against the pathogens, right? So, antibiotic resistant pathogens can be easily killed, can be easily be tackled by discovering different and novel antibiotics through these microorganisms. Industrial enzyme applications. Now, these microorganisms are also used for industry production, industrial enzyme production. Microbial enzymes with extreme stability. So, you can use these microorganisms to produce the stable and heat tolerant, heat stable enzymes that can be used for various industrial processes where the high temperature is required, for example, for the food production, for the many other applications. Microbiome medicines. What is the microbiome medicines? Microbiome medicine is something which you can use based on the whole metagenomic or microbiome analysis. So, to understand how microbial communities composition affect the human health, for example, gut microbiome, oral microbiome, vaginal microbiomes, and accordingly, precision medicine can be thought of to develop against that particular microbiome carrying individuals. Now, I just give you an example. Oral microbiome affecting the pregnancy outcome. It seems to be very awkward, but yes, it is. A lot of literature is available on that aspect. So, if you control the oral microbiome, you can control the pregnancy outcome. You can control the stillbirth, you can control the premature baby, you can control the pregnancy deformities and so on. So, this is how the whole microbial biome to understand and to design medicine for that. Not only exactly the pills or drugs can be designed, but I think the dysbiosis can also be controlled. You can maintain the good bacteria, right, and exchange the bad bacteria from that particular microbiome, maybe gut, maybe oral. Probiotics can also be used for that. So, all these things can be easily handled if you understand the whole genome analysis of this. Now, say for example, one of the case study, Indian microbial diversity hotspot, rich soil microbiome. Indian agriculture soils harbor exceptionally diverse microbial communities, particularly rich in bacilli, pseudomonad, and actinomycetes, and these groups contribute significantly to the soil fertility, and this is one of the advantage. Plant diseases suppression and nutrition recycling in topical and subtropical environment. So, to understand the whole microbial world through genomics would be easy to identify individual microorganisms from these groups and then to use further for the purpose of the good or rich soil producing the fertile soils and so on. Traditional fermentation system. India's rich traditions fermentation relies on the complex microbial consortia that have been maintained for centuries, from idli, dosa, fermentations to pickle, preservation, and these traditional processes showcase the practical applications of the microbial diversity in food productions and preservations. So this is another advantage. Industrial biotechnological potential is again, in industrial biotechnological potential, you have ongoing metagenomic projects, across the Indian ecosystem, continue to reveal novel microbial taxa. You can come up with a new microbial taxa with a different characters, different phenotypic expression, different genotypic expression, which could have a significant industrial potentials, and that can be used in future for various enzyme productions, various food productions, and so on. Many of these newly discovered organisms produce the unique enzymes and metabolites as a byproducts, or metabolites, are byproducts of the bacteria. Sustainable for pharmaceutical, agriculture, and industrial application. So these are some advantages which can be taken into consideration by understanding this microbial flora understanding. So keeping that in view, what should be the future direction, and how we can have in future look into the microbial diversity, and to know the microbial diversity and to use the microbial diversity for the benefit of the mankind. Now what we can do, we integrate the multi-omics and big data analysis. Now this is the new concept. This is the age of AI, artificial intelligence. So what you need is a big data. We have a huge data available on the microbial diversity, on the microbial world, where we can have a number of microorganisms, both in bacteria, viruses, and fungi, protozoa. Now, how to integrate the omics technology? How to integrate the different omics like genomics, transcriptomics, and proteomics, and metabolomics, and to integrate. Now, I'll just pick up the first genomics. Genomics means you can have the whole genomic data, and this genomic data will tell you how the diversity of these microorganisms are there, how you can separate these microorganisms, what are the different genes, different features, different characters are present in these different microorganisms. You can identify it. Whole genome you can identify it, right? Then you can also look into the gene expression part. What are the different genes are expressing in one individuals? What are the different genes which are not expressing in one individuals? And that will also give you the idea about how these genes express and how these genes are utilized, right? And how these genes are, for example, transcriptomics. In the case of transcriptomics, you can look into how transcriptomics will give you the precisely specific gene expression which are not expressed in other individuals. So you can pick up these individuals. So this is one of the advantages. You can also understand into the functional genomics. You can understand the functional genomics of a particular individual genome, and there you can know what the function of these individuals in the future. Proteomics is another area where in the proteomics you can find how the whole proteomes, which is the outcome of the genome, right? Genes to protein, right? As a central dogma, genes, DNA to RNA to proteins. Now, genes to proteins means like gene product, and what kind of gene products are being produced, right? And how these gene products can be utilized for the future, for individual microorganisms, as well as from the microorganisms can be utilized for your own benefit of human or for animals or for plants. The metabolics is basically the small molecules which are produced in the bacteria during metabolic processes, and this is the this is again the very small molecules, and these metabolomics can be identified, can be studied through the techniques we call LCMS. Now, LCMS give you the idea about how these metabolites can be identified. Now, individual microorganisms, even within the species means like E. coli may have like 6,000 different strains, and each strain may have a different character. So these metabolic characteristic features, or these metabolomics through LCMS, will give you the clear-cut picture that how these individuals vary. So this is one of them. Now, whole data, that is genomics data, then you have a transcriptomics data, and the proteomics data, and the metabolomics data comprehend together and give you a model we call AI, a machine learning model. That models can be used for prediction, estimation of metabolites, utility of these particular microorganisms based on their ecosystem, based on their different backgrounds, and one can easily make use of these individuals having the characters identified through all these techniques, and you can integrate this AI model through machine learning models and give you better picture of the individuals to understand about it. Toward a unified microbial species concept. Unified microbial species means we can have a genetic coherence, that is a species defined by genomic cohesion, recombination pattern reflecting actual gene flow within the population, ecological specialization, recognizing the particular species based on certain characters, based on presence in ecological state, conditions, distinct populations, right, as a taxonomically significant unit, even when genetically similar. So sometimes a species may be genetically similar, but having even the similar patterns of a species, you can have a different ecological patterns, different ecological state. This can be identified from a species level. How it evolve? What is the evolutionary patterns of this species? It emphasizes the lineage over a period of time on evolutionary scale, which independently evolved through mutations, regardless of the arbitrary genetic similar threshold. Doesn't matter whether the species are the same or not, but over a period of time it might change due to the mutations and due to the changes in the different species, and that changes basically help us to understand what kind of a different species we have, even the same ecosystem or in the same niches. Conservations and ethical consideration. Microbial conservation means protecting microbial diversity, requires preserving entire ecosystem, as most microorganisms cannot survive outside that ecosystem. So you have to provide, you have to maintain that ecosystem, proper climate conditions, proper habitat, and then pollution threat is also one thing which can, some pollutant can also destroy these species and species might lost. So all these issues are there to maintain that species within that ecosystem. Now once you characterize a particular individual species in one ecosystem, it has to be conserved. This is what we call the ecological or microbial sustainability. For that you have to maintain. So there's a lot of issues where you might lose that strain, you might lose that species, and in that case the conservation is one of the issue. Responsible bioprospecting. Now developing framework for ethical exploration of microbial resources, ensuring fair benefits, sharing with the communities and countries where the valuable microorganisms are discovered. Now, this includes the respective traditional knowledge and ensuring equitable access to biotechnological benefit. How these species, these different strains specifically make use of these species into the different industrial application, different biotechnological product. So this is again one of the aspect for this conservation of this species. Now international collaborations, establishing global initiatives for microbial diversity documentation and conservation, recognizing that microbial ecosystem across the nation, across the globe boundaries, and require coordinating international effort to understand and protect. So the species which you identified in India and the same species should maintain all across the globe with the characters, with the features, genetic and phenotypic, so that the stability and predictability model can be maintained. So the bottom line and the take home message is that you can predict and you can, having the stable character, you can predict the particular individuals under particular species or some order or phyla or family, basis of the character that you conserved, right? So it should be internationally similar, conserved with the collaborations, making the documentations through form of publications or through form of documentation in the book or manuscript or so on. Now, what we conclude finally is the embarrassing microbial diversity for science and society. Microbial systematic stands at a revolutionary crossroad where the classical methods merge in cutting edge, and these classical methods which are used for the process of characterization and maintaining these microbial world in a particular hierarchy can be further understood through microbial diversity, and that can be make use of future as a functional genomics, using functional genomics as a future make use of these microorganisms for various applications, various biotechnological applications, industrial applications, and many other things. So, in a nutshell, whole microbial world can be studied, can be categorized, and keeping them into different places, ordered systematically, so that whenever you need, you go and pick and use. So, ultimate purpose of this classification, ultimate make use of this purpose of this classification, what is the use of this classification is that you can easily handle these microbes for the benefit of mankind, for medical production, for medical benefit, for agriculture benefit, for for industrial benefit, or for many other use of these microbial flora in different categories. With this, I thank you very much for these lectures, and I hope we'll meet again for the next one. Thank you.